Good morning students, I am Shomajit sir. Today I am here to deliver my lecture on topic DNA, that is deoxyribonucleic acid. So this is from the chapter molecular basis of inheritance. So without any delay, let's start with the topic DNA. So let's start with the topic DNA. This is from the chapter molecular basis of inheritance. So first we know that nucleic acid, there are two types. One is DNA and RNA, and these are the building block of genetic material. So DNA is the genetic material of most organism, and DNA is the genetic material of human beings, you can see. And RNA, okay, RNA is the genetic material of some viruses, and RNA mostly functions as messenger. All right, so RNA mostly functions as messenger, and DNA is the genetic material of most organisms. And viruses, like some viruses have RNA as genetic material, like coronavirus. Now let's see the structure of DNA. So polynucleotide are the polymer of nucleotides, that is DNA and RNA are polynucleotides. You can see the phosphate group is attached, nitrogen base is attached, pentose sugar is there. You can see nucleotide 1, nucleotide 2, nucleotide 3 and nucleotide 4. Now let's see the structure of polynucleotide chain. Okay, so polynucleotide are the polymer of nucleotides. Okay, DNA and RNA are polynucleotides. So you can see in the structure, one is phosphate group, that is P, nitrogen base is there. First one you see the diagram, that is thymine, and the sugar is there. This comprises of a nucleotide. So nucleotide constitute nitrogen base, pentose sugar and phosphate. So again I repeat, nucleotide constitute nitrogen base, pentose sugar and phosphate. When nitrogen base and pentose sugar is a nucleoside, okay, so that means overall nucleotide is equal to nucleoside plus phosphate. Okay, and in the pentose sugar there are two types. One is ribose in case of RNA and deoxyribose in case of DNA. Okay, and in the nitrogen bases we have two types. One is purine and pyrimidines. So here you can see the nitrogen bases. These are of two type. One is purine, another is pyrimidine. Okay, so under purine you can see adenine, guanine are there, and in pyrimidines we have cytosine, thymine and uracil. So all the structures are given, you can see. And thymine, okay, you can see cytosine, thymine and uracil. So we know that thymine is present in DNA and uracil is present in RNA. So you can see the structure of a polynucleotide chain, that is DNA. So nitrogen base is linked to the pentose sugar through a N-glycosidic linkage to form nucleosides. Okay, so a nitrogen base is linked to the pentose sugar through a N-glycosidic linkage to form a nucleoside. So you can see the structure, pentose sugar, and when attached, pentose sugar is attached with nitrogen base, that bond is N-glycosidic bond, the bond that is attached with the carbon 1. So next you can see the differences between nucleosides of DNA and RNA. Okay, so in RNA you can see adenosine is there, guanosine, thymidine and uridine, whereas in case of DNA we have deoxyadenosine, deoxyguanosine, deoxycysteine and deoxythymidine. All the structures are given, you can see. Next you see the structure of polynucleotide chain. It consists of nitrogen base, sugar, phosphate group. This entirely three combine to form a nucleotide, that is deoxyribonucleotide. So all the figures you can see for a nucleotide, center one is a pentose sugar. In the fifth carbon atom that is attached is a phosphate group is attached to the fifth carbon atom, and in the first carbon atom nitrogen base is attached. Okay, so pentose sugar has a 5 carbon atom. So you can see in RNA each nucleotide has an additional OH group at second position of ribose. Okay, and two nucleotides are linked through 3 prime 5 prime phosphodiester bond to form dinucleotide. When more nucleotides are linked, it forms nucleotide. So you can see in the ribose sugar, OH group is present in the second carbon atom. Okay, and in case of you see in the right hand side in polynucleotide, you can see one chain is attached with another chain. You can see phosphodiester bond is there. Now DNA, Friedrich in 1869 identified DNA and named it as nuclein. So Watson and Crick proposed the double helix model of DNA. It was based on X-ray diffraction data produced by Wilkins and Franklin. Now the scientist's name and pictures are given. Now in the structure of DNA is made up of two nucleotide, that is polynucleotide chain. Okay, so DNA is made up of two polynucleotide chain coiled in a right-handed fashion. You can see the pitch of the helix is 3.4 nanometer, that is 34 angstrom, and the number of base pair in each turn is 10, and the distance between adjacent base pair is 0.34 nanometer. Okay, that is 3.4 angstrom. So you can see in the left hand side the diagrams are given. One curve, you can see the diagrams. Next you can see the backbone of DNA is formed of sugar and phosphate. Okay, so the backbone of DNA is formed of sugar and phosphate, and the bases projects inside. Okay, the two chain have anti-parallel polarity, that is one chain has polarity 5 prime to 3 prime and another has a polarity 3 prime to 5 prime, so they are anti-parallel. Okay, so you can see in the right hand side one strand is 3 prime to 5 prime, another is 5 prime to 3 prime. Okay, so you can see the structures given, the major groups, the minor groups, the sugar phosphate backbone, everything is given. You can see the nitrogen bases. So you can see the bases in two strands are paired through hydrogen bond forming base pairs. So you can see A pairs with T, that means adenine pairs with thymine. It has a 2 hydrogen bond, and cytosine pairs with guanosine, it has a 3 hydrogen bond. You can see purine comes opposite to a pyrimidine. Okay, so this generates uniform distance between the two strands. You can see in the right hand side the diagrams are given. So this is the structure of DNA. You can see the phosphate group, the pentose sugar, and the nitrogen base. Alright, left hand side is the DNA, its magnified portion is given there, the right hand side of the picture. So in the left side you have a DNA structure, and the right side you can see the base pairs are highlighted. So next is the Chargaff's rule, is very important. So in DNA the proportion of A is equal to T and the proportion of G is equal to C. Therefore adenine plus guanine is equal to thymine plus cytosine, or adenine plus guanine or thymine plus cytosine is equal to 1. So this is all about the Chargaff's rule. Always the purine is equal to pyrimidine. So you can see the DNA, that is a bacteriophage has 5386 nucleotide. A bacteriophage lambda has 48,502 base pairs. E. coli has 4.6 into 10 to the power 6 base pair, and haploid content of human DNA is or has 3.3 into 10 to the power 9 base pairs. Now you can see the calculation of the length of DNA. So you can see length of DNA is equal to number of base pair into distance between two adjacent base pair. Okay, so example, you can see the number of base pair in human is 6.6 in 10 to the power 9. So hence the length of DNA is 6.6 in 10 to the power 9 into 3.4 into 10 to the power 9 minus 9. So it comes 2.2 meter. And in E. coli the length of DNA is 1.36 millimeter, that is 1.36 into 10 to the power minus 3 meters. Okay, so the number of base pair is this by this. Okay, so you can see 1.36 into 10 to the power minus 3 by 0.34 into 10 to the power minus 9. So it comes a 4 into 10 to the power 6 base pair. So this is all about the calculation of the length of DNA. So thank you, students.